Melanoma Matters is an educational resource, and the opinions we express are our own and do not reflect medical advice. If you have questions specific to your own situation, please contact your own clinician. This podcast may be available in both audio and visual, so please check your platform.
Hi, everybody. Welcome to Melanoma Matters. I'm Sapna.
And I'm James Larkin. Hi.
Okay, so today we're going to discuss, we think this is the first time since we started doing the podcast that a drug has gotten approved, right? In the kind of 2 years we've been recording?
Yeah, because the last one was lifileucel. February 24, FDA.
Oh, and we had a bet going about that. Remember the bet?
I've lost it because
you've lost the. Okay, go on.
No, I think it was about regulatory approval in the UK.
It was
date of, and I hoped it would be by May 26. Or maybe
no, 25. I think you were even like May 25 because it was February 24. It was approved.
Yeah. And now we're in August 26, and we don't have a decision even.
But it's gone. It's gone for review, right?
It's being reviewed. So I hope there'll be a decision Q3 fall, as you would say. And then we have a separate reimbursement. But the decision on reimbursement won't be this year. That'll be already next year.
So none of that is the topic of our podcast today. Our topic is the next drug approval that has been approved. And my quick fire to you, James, in your poshy British accent. How do you pronounce this drug name?
I've got no idea.
Okay. Too easy an answer. Come on, give it a try.
No, because normally what I do in this situation is I find a video of Kevin Harrington on the internet.
Shout out Kevin Harrington. I was looking to see if I could even put it into like a pronouncer thing, but it's too new a word in the lexicon. Okay. Tudriqev. Don't invert the R and the D.
Tudriqev.
Tudriqev.
Quev, not Kev. That'd be Kev.
Kev, I think, because there's no QU. I think it's Kev.
Oh, Tudriqev. Tudriqev.
Okay, so which syllable do we need to put the accent on?
A third one, I think, don't you?
Tudriqev.
Yeah.
Or Tudriqev. Yeah, let's see.
Tudriqev. Tudriqev.
Tudriqev. Okay, good. So, okay. So we're talking about Tudriqev approval that got FDA approved just recently here in August. Any thoughts, any idea if it would go in front of MHRA?
Yeah, I presume it will. But who knows? And I guess the discussions will be similar, and maybe broadly some of the discussions are similar to the discussions that we've been having about lifileucel. Non-randomized trial, anti-PD-1 refractory population in melanoma, even contribution of components, if you want to stretch that far, which has come up a little bit with lifileucel. So it's almost as if there are a lot of the same themes, with the addition in this situation of these questions of injected versus non-injected lesions, and our special question, which is that if we have access to these drugs, which you do and will, other things being equal, how would you choose because this is the big question. If you've got a patient in clinic, you've got two quite different strategies here. Do you sequence? How do you make a choice?
Yeah.
So, go on.
Maybe it's also worth bringing up at the end. We'll bring up what you just said, if I can remember it. Maybe I should write it down. Okay, so I like everything that you said about contribution of components or contribution of effect, and then non-randomized. These are studies of approximately the same size, 140, 150 patients. In lifileucel, just to clarify, the contribution question was always about how much the IL-2 was doing versus the TIL, because IL-2 is a known regimen. But very similar to this conversation, it's thought that that sort of known entity is contributing very little in this patient population. It's certainly not contributing to the bulk of the effect. So this is based on the IGNYTE study, which was an episode that we recorded, published in JCO, Mike Wong, 2025, I think.
Yeah.
And which episode was it? Do you remember?
You know that I don't know that.
Yeah, fail.
Another fail. 2 fails already.
No, we'll fact check it. It's one of our recent ones. So based on that publication, of course, they had gone to the FDA and received a complete response letter around June or July of 2025. I think it might have been June. And then they met with the agency and said, we've got questions, can you help us understand this kind of denial? They got a second review in April and got a second complete response letter. And then really like a phoenix, it feels like they were kind of saved from the ashes and got a third review. Now, to the best of my knowledge, this is the only time we've seen the public kind of like an open discussion. So the discussion in front of the Cell and Gene Therapy Advisory Committee at the FDA was live streamed on YouTube. You can watch it. I'm not aware that those other reviews have any sort of publicly available viewing or discussion, but this Cell and Gene Therapy Advisory Committee was made up of about 13 people or so, 4 dedicated melanoma oncologists. We could say their names, but you could also just see who it was on the video. And it's a 7-hour video, and much like the ODAC for OGN, I have sunk many hours of my life watching this and rewatching and playing it. That feels sad, right? That I've Netflix filled with this information. But you and I have said this before, and sometimes I say it to myself to reassure myself. I actually do spend a lot of my life and time and brain power thinking about what we do and our treatment and our landscape and how to, I really do. For better or worse, spend a lot of my time thinking about this. And so on my free time, actually we'll go deep into the Cell and Gene Therapy Advisory Committee broadcast. And I've watched it a few times. We watched parts of it together. So we have decided to just go through maybe some bits of it. Does that sound fair?
Yeah. And before we do that, so why the live stream for the third meeting? Do you know?
I don't know. I find it helpful. I thought the advisory committee made lovely comments, both from our melanoma colleagues, as well as the non-oncology advisors, as well as the FDA, as well as the experts that the sponsor brought. I really think it does help you not just see things from one perspective when you can see the whole thing like that. Who has 7 hours of their life to watching this thing over and over? I don't know, but I do think it's useful. There were some patient voices on there. There was a patient representative who asked some really good questions about the age. She said, I think it looks like to me the majority age was under age 65. How do you square that with how this might work in a patient population older than that, or with more comorbidities? And the sponsor reply was like, well, it was just, here's our median age, and 59% of patients were under 65, but we had no restrictions on enrollment. That's true. That's a factual answer. But I thought that patient representative's question was really well taken, and you could kind of see why she was asking that. Anyway, let's maybe get into it, shall we?
Yeah, sure.
Okay.
Kick us off.
So I think one of the main sticking points that the FDA had in their first reviews that they needed clarification on was, is RP1 plus nivolumab, Tudriqev plus nivolumab, is this a local therapy or is it a systemic therapy? We're using it in a patient population that has progressed on PD-1. Maybe we start there. How certain are we that this is a PD-1 progressing population?
Well, I mean, we talked about this when we talked about the paper. And so it was pretty tightly defined, adjuvant or metastatic. I don't think, maybe we'll talk about this at the FDA meeting. So in the paper, they didn't use SITC criteria, right? But then they did actually go back and then reanalyze using SITC refractoriness criteria. So I think it is a refractory group, but the question is always going to be in any refractory group, even if it's been well defined and the trial's been done well, what level of efficacy might you expect from continued anti-PD-1 therapy? And there was quite a bit of discussion of that, right? You've watched more of this than I have. And it feels like most people thought it was around, let's say 5 or 10%, I think, which feels about right. But go on.
Yeah, I mean, I think the real conversation that they tried to highlight here was you really weren't allowed on this study if it could have been pseudo-progression. They really wanted you to be progressing, and that progression needed to be essentially double scan confirmed, right? You couldn't have, except with maybe the exception of if you were coming off of adjuvant PD-1, because you don't really need to double confirm somebody's brand new metastasis that you can then biopsy prove. If you were coming off of PD-1 based therapy and you started to have some progression or new lesions developing, they just in general wanted that confirmed so as not to kind of eke out any ongoing PD-1 monotherapy effect. So I think the advisors felt pretty comfortable this was a true PD-1 refractory population and not subject to enrolling patients who might be pseudo-progressing and still getting a benefit from PD-1 mono. But maybe it brings up a question. We've talked about this before in trial design. We might have talked about it in the IGNYTE paper. Is the patient population progressing on adjuvant PD-1 the same as the patient population progressing on metastatic PD-1 based regimens?
Well, no, in a word, I think is the first thing to say. But on the other hand, I mean, maybe a way to look at this in terms of is from a patient perspective in terms of unmet need, whether you progress on an adjuvant or neoadjuvant for that matter regimen or with metastatic disease to anti-PD-1, it's a bad situation to be in with limited treatment options, particularly if BRAF wild type. So no, it isn't the same group of patients, but I think in terms of trial design and approval and everything else, it's a reasonable way to do it.
I like that. Biologically, there could be some nuance, but I like how you said from a patient perspective, it's the same. So back to this question, is it a local therapy? Is it systemic therapy? One of the key sticking areas in the discussion was, and you and I have talked about this, I call it Larkin-izing clinical trial data sets. I know that's not great. Larking sounds worse. Larkin-izing is what I'm going to say. Where we have a nice clinical trial data set, and now I want to reproduce that in my clinic or even do better in my clinic. So how do I look at this data and then look at the patients coming through my clinic and figure out who to treat? Maybe more importantly, with regards to this type of therapy, because we are deciding where to put the RP1, I need some guidance on if I should be aiming to inject 100% of a patient's metastatic burden, or is injecting 50% of it good enough? Or am I going to get some benefit even if I can only inject 10% because of this systemic effect? What burden of somebody's disease do I need to inject? That helps me pick out the patient population because we only get 10 mLs with each injection. Should we think of it differently? If you're injecting 100% of the disease versus injecting part of the disease, do those feel like separate calculations of benefit or response? The FDA felt this way, and they essentially excluded certain patients and redid some response calculations.
Yeah, there's lots of questions in there. I don't think I know the answers to any of them either. So the fundamental thing, I think, is this just a local treatment, or is there a systemic effect? And I think it's been shown fairly clearly that it's both, and you can get a systemic effect. And they showed that at the FDA meeting, I think, fairly convincingly. Very difficult to read that graph right because there were so many vertical lines, but nevertheless, there clearly is shrinkage of non-injected lesions. But then, if you've only got 10 mils, where'd you put it? And we spent some time talking about that as well. And
10 mLs a lot.
I don't know. I've got no idea the answer.
Do you know what the max volume is for T-VEC?
No, but you do.
4 mLs.
Wow.
So it's
but I mean, I think it's worth saying that when they analysed the, let's call them 100% injected versus not 100% injected, and there were quite a few patients of the responders who were not quite half, but you're pointing at my chin. I don't know why.
Can you get close to the microphone?
Oh, I see. I was relaxing too much.
Yeah.
It's the very hot weather. So the ones, a lot of the responders had 100% injection. So then it feels to me that if you've got someone in clinic where you've got disease where you think you can inject most or even all of it, this is a pretty good option, I would have thought, for several reasons. One, that what I've just said, we know that it's effective locally. Second one is maybe that kind of patient who's progressing almost by definition with relatively small volume disease, often superficial, let's say. This is a good option in terms of toxicity, because I'm thinking about lifileucel now, aren't I? And I'm thinking that's a treatment that, if you look at the toxicity profile, is really pretty different from this. So that's what I'm thinking. But then the kind of bigger question, in a way, because we've had T-VEC for quite a long time,
10 years,
and it's worth mentioning Kevin Harrington again. Kevin, my colleague, has done a lot of work, and he was at the FDA as well. It would be a surprise if we didn't see efficacy with this treatment. And obviously the idea biologically is that you've got greater efficacy than with T-VEC. And we've got a sense, haven't we, I think, already about how we want to use T-VEC. But let's say we're going beyond the sort of superficial loco-regional type of disease, small volume, and you're getting into visceral disease. Do you inject it? Do you not inject it? I don't know the answer to that. And I still don't have a wonderful feeling, honestly, from reading the paper and listening to all of this about those kinds of patients who are in that group. Do you inject all the superficial stuff and then not use any for visceral? And then, you're obviously hoping for a systemic response, or do you mix it up a bit? I don't know. I mean, you've got more experience with this than I have, I think.
So I think we teased this on the IGNYTE podcast towards the end, where, definitely don't just rely on bystander for the deep. There seems to be some benefit in injecting those viscerals, but we still have to kind of feel that out, right? It's really unclear in that superficial and deep, even, it's really unclear. Are we talking about splitting a 10 ml dose, or are we talking about doing 4 injections into superficial and then rotating and doing 4 injections into deep? So it's a little TBD, and some of this will come out in the phase 3 study, I think. But
how about in clinic, if you've got someone in this kind of situation? In other words, they've got some loco-regional disease and some visceral disease, and you can't inject everything anyway because 10 mils isn't enough. And you're going to leave some superficial stuff, right? Because you're going to decide to inject some deep stuff. Would you leave your most lifileucel suitable lesion, 2 centimeter lymph node, 2 to 4 centimeter lymph node, not inject it. Obviously, hopefully the patient will respond to this treatment, but then if they don't, you've left behind the thing that may be most suitable for lifileucel or TIL generation as a subsequent line of therapy. I mean, that's very pragmatic, made up way to do it. But I'm just trying to think again about how to do best for the patient, actually.
I like the way your brain works. I will tell you, I'm going to be careful about how I say this, but it is not for every patient. It is for a uveal melanoma patient, but it is not for every cutaneous melanoma patient that I'm always thinking 2 and 3 steps down the road. And the reason is we do have a cure fraction with some of our therapies. And so I don't want to be playing defensively for plan B and plan C if plan A could work. And the only reason I say that with regards to this data set is some of the striking data that was shown in the FDA presentation was that in the responders, they have 47 responders. It's an approximate mixture, half and half, of patients who had all of their metastatic tumor injected and half who had some of their metastatic tumor injected and then uninjected. In those 47 responders, all bundled together, the overall survival was really long. It was 2 years plus, and these are pre-treated patients. These are not first-line patients. You know, whether we call that progressing on adjuvant or metastatic progression, it's a mixture of both. It is, what I wouldn't want to do is hold back on injecting everything if that gets you the highest response rate and the longest survival, because I'm thinking, oh, I need to get ready for plan B, because maybe you could have cured them in plan A with plan A. But I like what you're thinking, and you're not wrong to think of it that way. I think that's exactly right. And I think we all who do mostly melanoma predominantly, I think you have an instinct about a patient after you do a first few injections, whether it's T-VEC or RP. You know if this is likely going to be on a good trajectory or you need to be getting yourself ready for something else. And so, yeah, it goes back to, should you leave something? Should you inject it all? If you're trending towards a response, should you go ahead and add in that last little bit that you didn't inject? Because that does seem to give you the highest response rate. If we're being clear, injecting everything seems to get you the highest response rate. And we'll get to tumor burden here in a second, because there was a nice graph on that. But a question I was going to stick to the end, but I'll ask it to you now because you just sort of brought it up. One of the things we're debating, and we asked it we had our inaugural Colorado Oncology Skin Cancer Conference. We called it the Peak Perspectives in Cutaneous Oncology. We had that last weekend. We had great, local and national and international speakers. But one of the conversations we had that we all agreed there's no data for. My TIL nurse ran a session, and she ran a session with our TIL surgeon about TIL and procurement and then just the flow of patients, et cetera. And then she said, well, I'm just going to use this expert panel to ask you guys a question. Can we do a harvest from a lesion that's been T-VEC'd? And when that question came up to me in clinic, I had said I wouldn't. If you're telling me you're going to T-VEC and then 2 weeks later take that same tumor out, it's not my favorite. I'm a little concerned. But on the other hand, it's not like you're radiating it. It's not like you're giving chemotherapy. You're not killing the T cells. My concern goes to a little bit of what Kevin Harrington discussed at the FDA. Kevin discussed some of the work I think that Lizzie Appleton and you guys have done there in the mice, which is when you make a mouse model, immunocompetent mouse model, and you put melanoma tumors in the flank and you inject one with RP1, I don't know if they did it with Nivo. I think they did it with Nivo, but systemic Nivo. The population of T cells that you get in that injected tumor, you get CD4 and CD8s, but you get two different populations. You get a virally enriched T cell population and you get an anti-tumor T cell population. And then in the uninjected tumor, you get that nice, optimally, hopefully migrated batch of anti-tumor T cells, but you don't get this viral T cells. So my concern to my team was, look, if you harvest too soon after a T-VEC, the T cells you might expand outside the body, they might just be your viral T cells. They may not be your anti-melanoma T cells. And then Mark Ferries made a good point at that panel. He said, yeah, but that's already happening. We already have viral, experienced T cells on the skin in every melanoma tumor. And when you ex vivo expand, you're already enriching for some of those. But my fear with T-VEC is you're really, that would be a question for Kevin. In proportionally, proximally after an injection, is it, I would think, right? It's more viral T cells in that proximal time period, and then maybe it switches later into more anti-tumor T cells in that injected tumor. But we asked the panel, and rightly, nobody had any experience here. I wouldn't suggest it. I wouldn't, promote that, but it's something that's coming up here because we might end up having to do it. But that gets to your question, which is, should you save something for TIL, or can you ex vivo expand from an RP1 Tudriqev tumor?
I'm feeling conservative, I have to say, about like you, because the data that we have for lifileucel is based on non-virally injected tumors. Mark Ferries is right as well, but I agree with you. I mean, it's one thing being virally experienced, it's another thing having a load of virus directly injected into a lymph node. And just from the kind of efficacy question for this type of therapy, let's just say you've got someone who's got quite a lot of subcutaneous locoregional lymph node type disease. I'd be pretty happy injecting a lot of it, but leaving one quote unquote juicy lymph node, for want of a better term, and then if you've still got some left over, injecting your metastatic disease. It doesn't feel to me that doing that kind of strategy and missing one out from the chances of it working, okay, yeah, maybe it might have some detriment, but I'd be quite surprised.
And if you're saying of 8 injections serially, leaving one out and then catching up with it later, or what were you saying there exactly?
Yeah, just deliberately in this situation where you've got obviously in someone who is potentially suitable for lifileucel as a subsequent therapy. Let's be clear about that as well, because there's no point doing it otherwise. Just being maybe a little bit conservative about how you start doing your injections. And I wouldn't say never inject that. I mean, if someone's obviously responding to this therapy, then you'd probably go all in. Right? Yeah, exactly. But just when you're not sure about that, I don't see a strong, in the absence of data, this is us speculating, right? I don't see a strong reason for just sort of keeping a lymph node, let's call it that, in reserve, in case you quote unquote need it in the future. But I mean, listen, we're going to get some experience of this, aren't we, in clinic? Go on.
And that's what I mean. Like, after you start doing a couple of these, you're going to get a handle on whether it's working in the injected tumors or not. And then to your point, if you need to save that lymph node, you save it. But if it looks like it's working effectively, you might then add in that lymph node as another injection site. So maybe we get to 2 more things and then try to tie this up. Let's talk about the percentage of patients who had prior adjuvant therapy. And then maybe we want to talk about maybe a tiny bit about this math of injecting everything, not injecting everything, what the response rate looks like. Or we could talk about tumor burden, or we could try to do all 3. You pick.
Yeah, so the math is complicated, right? We can agree that. We'll save that till last, right?
Okay, let's save that for last. Yeah, okay.
But the waterfall plot maybe that was shown at the FDA meeting is pretty illuminating, isn't it? Because it was sort of mapped in a non-standard way to start off with, because usually with these things it would go from smallest to biggest.
It's figure 2D in the paper, and I think when you and I looked at this paper, we didn't even pay attention. I think I now I'm remembering saying, I don't even know what this is graphing. I don't understand this. And it's super small, and so you constantly have to like zoom to even understand it. And I didn't, but now that I've seen it presented at the FDA, I understood what they were presenting. I would have preferred they graphed it in that traditional waterfall plot though, which is response least to greatest. Here they plotted it with tumor burden least to greatest.
Yeah. So I mean, I don't have it in front of me, but I can see it in my head. So one thing to say is there's quite a lot of patients really with a very small burden of disease,
less than 10 centimeters.
Yeah, and even quite a few 2 centimeters or left.
Yeah.
at that left-hand end. And
Yep.
quite a few of those were adjuvant, denoted by a plus at the top of the column. And then if you look at the other end, where you've got 10 centimetre or greater burden, surprisingly, just around that, I think from about 8 to 10 centimetres, there are also a cluster of adjuvant patients, which neither you nor I could really easily understand, that volume of disease from someone who's coming off the back of adjuvant. So I guess this is all anecdotes and we're talking about individual patients, but I couldn't really understand that very well either. But I think, and this is the responders, obviously, I think it's fair to say that a lot of them did have a tumour burden that was under 10 centimetres overall. And getting back to your question, which was about what proportion of this disease volume has been injected and how does that correlate with the response, or otherwise I'm not sure I've got a clear answer to that in my head.
I think Paul Chapman asked that on the meeting. He said, tell me what proportion of your complete responders were in patients who had everything injected.
Yeah.
And the answer was, we'll get back to you after the break.
And did they?
We will fact check that.
Okay.
Let me write it down.
So did that cover points 1 and 2? In other words, everything except the math, or not? I'm not sure.
It was tumor burden and it was prior adjuvant. So I bring it up just to say this starts to help me Larkin-ize my clinic. Okay, somebody progressing on adjuvant therapy with kind of oligo disease, maybe TIL is, well, I wonder if this is going to suffice for that requirement of TIL that they've received prior checkpoint inhibitor. It should, if they progress on this. Anyway, it's that adjuvant patient with kind of low volume, definitely under 10 centimeters, total tumor burden. We think that's what that was. It's hard to know if that's total tumor burden or target lesion tumor burden, but we think it's probably total tumor burden. Again, very surprising, the left-hand side of that graph. I don't think anything about the LDH because some of even the examples they showed didn't track for me. Some of those patients shouldn't have had elevated LDH. And so that LDH that is elevated is not likely due to their melanoma biology. It's due to something else. So I'm just trying to like refine in. So yes, okay, I think I can understand more the patient I might try to pick out for this. But let me ask you this controversial question. Somebody progressing on adjuvant PD-1, we have this data set now where we could just look at the adjuvant only patients. It's about a third of the responders. I don't know what percentage it is of the 140 patients, but of the 47 responders, it's about 15 of them, adjuvant only progression. We could calculate the response in them. Again, we'd have to know the denominator of what denominator of those patients entered, but it looks like this is a therapy that could work for them. What is your estimation? Do we have any data set? Cemiplimab, fianlimab, that 113 patient cohort that had received prior adjuvant or neoadjuvant. Do we have any data on how Ipi-Nivo or combos work, PD-1 LAG-3 work after when you're progressing right off of adjuvant therapy?
I'm not sure, actually. Honestly, is the answer to that.
Same. So my question in my head is, I've got somebody progressing off of adjuvant PD-1. Do I want to BRAF-MEK them? Do they qualify for lifileucel? I don't know. Do I want to CTLA-4 PD-1 them, PD-1 LAG-3 them, or PD-1 Nivo PD-1 Tudriqev them? Cool that we have
yeah, it is. I mean, I have to say, if someone's progressing off adjuvant treatment, anti-PD-1 outside trials, by definition, most of the time, I've said this to you before, they'll have small volume disease because you're scanning them a lot. and so I'm going for Ipi-Nivo as my first choice systemic treatment outside clinical trials and outside patients where you might want to go for some sort of other strategy. And then we could debate and discuss what the benefit level is there for Ipi-Nivo in that situation. Go on.
What is your gut telling you if that's how you're treating them in clinic? What's your gut telling you? It works half the time? It works a third of the time?
No, not as much as that. So I mean, you know how simplistically I think about this anyway, that if you've got someone who's truly anti-PD-1 refractory, 100% refractory, for want of a better term, and you're using ipi/nivo, they're going to have loads of anti-PD-1 on board already. So you're giving them some more anti-PD-1, but actually the ipi's doing the work, isn't it? And so then I'll just go back to the very first principles of what does ipi look like. And we know that in kind of a broad group of patients with advanced disease, the ipi response rate's about 20%, something like that, we could discuss. But if you've got small volume disease, normal LDH, which you normally will have, no brain mets, not on steroids, good performance status, actually it's north of 20%, I would say. And so I might be thinking 30-40% with the toxicity of ipi/nivo by definition. It's not ipi-mono. So that I think is a good option for some patients. What I was thinking about, though, when you were talking about who you think about for this treatment is recurrent acral melanoma, I have to say, because again, some of these patients, you can do surgery and all sorts of other things without getting into that. But this is a group, it's a big unmet need, especially in the leg, often small volume disease, often little benefit from checkpoint inhibitors, including ipi/nivo. And we know that T-VEC can work in this group. So it just feels to me that this is a natural group in clinic where you've got things that you might be able to inject all of it or most of it. And so that's where I'm thinking, actually. Go on.
Can I give you a cautionary tale here, though? I'm a little worried based on the label of FDA approval. The FDA approval, first of all, doesn't use the word metastatic. It uses the word advanced cutaneous melanoma. And you could almost argue there's going to be some insurance payer, at least in the US, who sees acral on the chart, on the medical record, and says no. So, but your point is well taken. And we said it in our very early Optum episode, where you said this actually is a great option for those in transit kind of acral. You've rarely seen a T-VEC not work for like a foot in transit melanoma. And I definitely have to agree with you there. So this would be a pity if we couldn't get it for our acral melanoma patients. But I'm a little worried about the label of the.
I think that would be a real shame. And whether I'm allowed to say this or not, I don't know. But I have to say that I think about acral melanoma as a subset of cutaneous. I don't draw a line between the two.
Exactly.
As far as I'm concerned, it's a subtype of skin melanoma, and it would be awful if that distinction was used to, I mean, maybe this is too strong, but I'm going to say it, to deny access to what I think is going to be a potentially effective treatment in a difficult to treat subset of the disease, actually. So that's what, yeah, go
on. Anyway, let's spend a couple minutes on the math. So the math is 25 patients of the 47 responders had all of their tumors injected. So had 100% of their tumors injected. The FDA therefore omitted them from the response analysis because they said the question at hand is not how well does your drug work when it's injected, how well does it work as a systemic regimen. So they excluded those. And when they excluded those and just used the patients who had some of their tumor burden injected, they removed those 25 from the numerator, from the 47, but they did not remove the 25 from the denominator, the 140 of the entire data set. And so Paul Chapman mentioned, I think you actually have to remove them from, if you're going to remove them from the numerator, you remove them from the denominator. That is true, but it's not even quite as simple as that. Those 25 patients, if you remove them from the denominator, are the patients who had everything injected and were responders, but you mean to also remove patients who had everything injected and were not responders. If you look at just the uninjected, the people who had injected and uninjected, those 22 patients, the response rate obviously is not as high as if you inject everything. Any more to say about the maths there?
No, but I think maths. But I think if you look at the second group where the response rate is lower, for me that confirms the efficacy, actually. And we talked a little bit about this before, because you could argue that there's a bit of a contribution potentially there from the nivolumab, blah 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 blah, but that isn't going to be above 10%. So then there's a delta, a bona fide delta there on the upside in terms of efficacy. So I think it's relatively, it's not relatively convincing. I think it's convincing, actually. And the biggest question is in clinic, which is where we started maybe, about how we're going to use this treatment. And I don't know, maybe I should ask you when you think you might have access to it. And then, as ever with these new treatments, I mean, I go back to the IPI mono 15 years ago, we learn a lot about them actually when we get access to them in clinic. Go on.
Yeah, agreed. A couple of things. Probably manufacturing. We're going to have drug supply in about 60 days here in the US. So I would think by ESMO of this year, we probably have a commercial supply. So we're thinking about how to get this, which really, really means playing nicely with our interventional radiology colleagues because we're saturating some of their capacity.
That was the main thing. But I mean, you saying that just made me want to maybe emphasize the fact that this isn't just going to be about oncologists, and it takes time, interventional radiology in particular. And I think it's really important for everyone to give their colleagues in interventional radiology a heads up that this treatment is coming, because what you'd hate is for it to be available and actually in terms of capacity to deliver, that it just wasn't there, and that's not great for patients. So I mean, anyone who may be listening to this, I would emphasize the importance of talking to colleagues now in the US and actually awareness ex-US as well. Yeah, okay, come on.
So, but I have two things to say, which is one, we've implemented here the first tumor board of the month. We do tumor board weekly. The first tumor board of the month, we're dedicating it now to our injectable patients. So we're doing the first 5 to 15 minutes of those cases, whether it's trial or standard of care. The interventional radiologist dials in or shows up. We review the scans really quickly. We either agree on all the lesions we've selected, or we let them choose if we're starting a patient fresh, or we let them change. If they say, look, you chose this one in the liver, it's sitting right next to the portal vein or right under the capsule, we can't do that. We give them a real say, because it is also very interesting to say that it's the medical oncologist choosing. And that may not, again, we've got 10 mLs to distribute. We've just had this whole conversation about superficial, deep volumes, et cetera. And we've completely neglected to say there's some mechanical and operational issues here too. We need to make sure it's accessible to our colleagues who are going to do the procedure. That's number one. Number two is IGNYTE-3 is still going. This is the confirmatory phase 3 trial. Patients progressing on a PD-1 based therapy get randomized to Nivo plus RP1 versus investigators' choice of kind of a menu of options. I think Opdualag being the most common option that we're probably choosing in the US. The question was, would we still want to enroll patients on that study when now we have access? We risk them not getting RP1 Nivo if in a randomized trial, and now we have it as a treatment. So probably enrollment's going to kind of plummet in the US, but we'll mainly move to overseas, ex-US as a predominant place. And you guys are doing it there at the Marsden, right? IGNYTE-3. So.
Yeah, and I'm sure that's right. And I mean, for the record, this is important, actually. I think that the primary endpoint of that trial is overall survival. And so we've all had all these long discussions about a single arm trial, injecting versus non-injected, contribution of components, et cetera, et cetera, et cetera. We're going to have a randomized trial, assuming that recruitment completes. It'll probably be a bit of time, but it's got an overall survival endpoint. And so then this will, I hope, put to bed some of this stuff in due course.
Yeah. Good. Well, congrats to the field. Congrats to Replimune. It's really exciting for us, at least here in the US, to have our hands on this now, be able to ask some more of these IRL questions. I mean, this is really the story of never giving up and believing in your asset. So exciting for them. Congrats to everybody. I think it's great for patients, great for us to have this option.
And it's a second approval for this class of drugs, right? So that's a big deal, I think. I'll mention Kevin Harrington as well, who spent most of his career working on this. So congratulations, Kevin, as well.
Kudos. Yeah. Okay. We'll do a tiny fact check on this one because there's some things we probably need to do if people can bear to listen for another 5 minutes. But we'll do it some other time. So thanks, James.
Bye.
Bye.
Hi. This is the fact check for the FDA Replimune discussion. A few things I think we talked about at the time that we thought we'd go away and check. So maybe I can just start as I'm talking, which was the date of the FDA approval for T-VEC. And you don't like me mentioning dates in general, but it was the 27th of October, 2015. So that was one thing. And then actually, maybe I'll just carry on, if that's all right, which was the next thing was which episode of Melanoma Matters do we discuss the IGNYTE dataset study that led to the FDA approval? And the answer is episode 81. So I will now hand over to you for the remainder of this fact check.
Well, a couple of things. I mean, that date of T-VEC approval is pretty telling for us. It's a benchmark. It's 11 years to the next oncolytic approval. So that's something. Telimagene, laherparepvec to vusolimagene, odirparepvec, vusolimagene, odirparepvec. It took me a while to learn telimagene.
I mean, listen, that was pretty good. I mean, I think only Kevin Harrington can beat that. Carry on.
Speaking of Kevin Harrington, you invoked him a lot during the episode, very kindly. I do want to make the point, he was one of the experts that was accompanying the sponsor at the FDA, but he was not remunerated for any of that. He did that all on his own dime. So just for conflict of interest purposes, he flew all the way from jolly old England to the FDA to, in support of this. So
wouldn't it be on his own kind of
pound? Pence?
I don't know, pence, but not dime. Definitely not dime. 10 pence. I don't know. Sorry. Carry on.
Well, it's 5 pence with the exchange rate. And then just the final thing I wanted to mention, Tudriqev. So we decided that's the name of the drug. We don't know where to put the accent, which syllable, but do you have it in front of you? Do you want to pull up the actual label?
I can try. It'll take me a second though.
Okay.
Oops.
Tudriqev. Package insert. God, I love the way you copy paste. I love when it comes up as Bing search. Old man.
But it's automatic, isn't it? I don't have any. I mean, they probably could change it, but it's institutional.
Mine doesn't. Mine comes out with like the whole URL. So something about your.
No, I prefer that. Actually, I don't like these kind of hyperlinks. But anyway, that's just a minor.
Yeah, I like that yours says package insert instead of like www. Okay, I'm gonna put a mark here. Okay, so you have the label. Maybe you want to read it off?
Oh, okay, yeah. So hang on, back to where I was before. Oh no. So sorry, 1 second. Oh yeah. Okay, so indication: a genetically modified oncolytic viral therapy indicated in combination with nivolumab for the treatment of adult patients with unresectable advanced cutaneous melanoma who experienced disease progression with a programmed death 1 blocking antibody-based regimen. Quite broad.
Well, not. It says advanced cutaneous melanoma. So that, as we talked about in the episode, might give us problems with acral, mucosal, even though many of us believe acral falls under the bigger heading of cutaneous. All it's going to take is an insurance company. So we may have to start writing in our note, acral melanoma subset, cutaneous melanoma subtype or something like that.
Cutaneous brackets acral or whatever,
Yeah, something like this. The other thing is they use the word advanced, not metastatic. So that's quite interesting. I don't know if we have another label that uses advanced instead of metastatic. And then the other thing of interest. So we spent quite a bit of time in the episode talking about objective response rate in the injected tumors, and as well as what's the response rate if you are not injecting everything. So go to table 3 in the prescribing information if you still have it open.
Yeah, give me a second.
I read this in a biotech newsletter this weekend, so I thought it was important to put in the fact check.
Oh, hold on. No, you might carry on actually. While I'm looking.
So the FDA has put in the label that the response rate is 24%. And so they subtracted out. Remember, this was the whole Paul Chapman math, and we were talking in the episode, you can't just take out the 25 from the numerator. You have to also take them out from the denominator plus
the other group of patients who had everything injected. So the FDA must have been able to sort that, and they took that denominator down to 91 and that numerator of 22 patients who had not everything injected had a response over 91 patients who had not everything injected. And so in order to prove this systemic effect, and so that comes up with an objective response rate of 24%, not the 33 to 34% that Replimune has talked about the last couple of years that's in the publication. Do you see it, table 3?
Yeah, well, actually, I'm looking at table 4. This is just from the FDA website, which is the summary basis, supporting document, summary basis for regulatory action.
Oh, table 4 in the prescribing information that you sent me is.
Yeah. And so table 4, efficacy results for the IGNYTE study. And it says applicant reported, evaluable patients 140, response rate 33.6, median duration of response 24.8. FDA analysis, asterisk 91 patients with at least one non-injected lesion. As you say, response rate 24%, median duration of response 14.14.
So that's the other important thing, is they took out that 24-month duration of response in the total population. So they've really, this is a different label than what the company probably, wanted, approved, and presented. It's down to 91 patients where you're not injecting everything, 24% response rate, 14.1 month duration of response. So just thought it was important to highlight that, because that feels, that's in the label now. There's no 33%. There's no 140 patients. It's just 91 patients.
Yeah. Very good.
Okay.
That's everything.
I think that's everything. Yeah. Anything you wanted to say about 81, the number 81, episode 81? You said something super geeky that I had no idea.
I don't believe that you had no idea, and I'm not sure how geeky it was, but 81 is obviously 3 to the 4th power.
Oh, obviously. I had no idea. And I said, what does that? He said, that's a unique number, 3 to the 4th. What? Had no idea. But as you say it, it's 9 times 9.
Yeah.
Great.
Good way to end.
Okay.
All right.
Thank you.
Take care.
Bye.
Bye.